let's get started. This is the final session for today's conference or meeting. So it's my great pleasure to introduce next speaker, Dr. Nathan Hedrick, who is a senior scientific director of clinical flow cytometry at BMS. Dr. Hedrick received his PhD at the University of North Carolina at Charlotte in microbiology and immunology. Before BMS, he worked at Pfizer, where he has led efforts in flow cytometry assay design, validation, and clinical implementation. Today, he's going to talk to us about some of the major stumbling blocks he has encountered in his work supporting global clinical trials and the strategies he's developing to combat them. Nathan?
Hey, thanks for the introduction there, Neethi, and thanks everybody for hanging in there. It's been a very informative day. I'll try to keep my comments brief today, about hour and a half. No, nothing. At least you laughed at that. The jokes seriously only get worse from here out. So what I'm up here to talk to you today about is what my lab does when we're not doing our day job. Of course, our day job is all about building assays, getting data for our clinical trials. And oftentimes what we would like to do is try to make our lives easier and also try to make flow cytometry more available to more clinical trials throughout the world. And to do that, we really focus on how can we beat these stumbling blocks that are present for our global clinical trials. Now, I'm Nathan Hedrick. I run the lab, so I get to get up here and talk about this work, but the people that actually did the work, many of whom are still here in the audience. I appreciate you guys sticking around. They actually get the job done. Feel free to bug them after the talk here. They can answer way more questions than probably I can. So what do we use flow for in a clinical trial? Well, I'm sure probably really kind of preaching to the choir here. But a lot of it is around pharmacodynamic assays. In some cases, we have some pharmacokinetics, depending on the type of drugs. This is usually done through deep immunophenotyping, some biomarker discovery, some safety. Probably the real bread and butter out of my lab is target engagement and receptor occupancy assays. All of these together, about 95% of the samples we run in our lab are whole blood assays, which means that these are taken from the patient, shipped out to either our lab or maybe one of our partner labs for analysis. So anybody that's ever seen me talk, I really like to pick on the audience. And I want to pick on you guys today. So, well, maybe not pick on you, maybe ask you a question. We'll just go with the question. So I mentioned stumbling blocks for clinical flow cytometry, flow cytometry in clinic. I know you don't have a mic, but you can scream it out. Anybody tell me what do you think one of those stumbling blocks will be? Nobody, not even my lab. Stability, yes, that's one of them. Thank you. Anything else? Guys, if I don't hear from my lab, end of the year reviews are coming up and they're going to be brutal. Just kidding. I'm just kidding. But yeah, stability is a good one. That's probably the first one we'll talk about. But really what the stumbling blocks we like to think about a lot in our lab are assay design. We don't try to poorly assay design. We try to avoid poor assay design. Assay execution, sometimes poor assay execution, instrument setup, harmonization. We've heard a good bit about that today already. Reagent problems, data analysis. That's, if I come back next year, you hear me talk, I'll probably be spending more time on data analysis. Problems with our samples and sample stability. To me, the biggest devil on this list is sample stability. And just so we're level set, when I say sample stability, what I mean is a time frame from where the blood is drawn from the patient to how long we have from that time it's actually drawn till it's actually unusable, till it gets to the point where the data is, there's such a difference in the sample that it's not no longer quality, no longer able to be read accurately by flow cytometry. And this varies. Some, depending on your analytes, depending on your portables, I've seen some as short as 2 hours. Some maybe 24, 48 hours on a good day. Some a bit longer. So why is this important? Well, sometimes it's not. Like, we see this a lot in some of our first-in-human trials, where you have your clinical site right next to your testing lab or close by to your testing lab. Of course, here, that huge dot would be my lab. We don't have a first-in-human site anymore. Used to, but this is great. You can get the samples from the patient to the testing lab very quickly. Now, you might get lucky, and you have a very long stability of your sample. And that way you can run a global clinical trial and bring it in, again, to my lab in BMS up there, from around the world. But what happens most of the time is not that. We usually have very short stability of our samples. What does this mean for us? Really, we have to test those samples in basically a regional basis, right? They don't have the ability to ship those, say, from Australia over to New Jersey or even England. Maybe in our lab in India, yes. We've actually proven that's possible. But you really need different testing labs. And this can be done, and we do it a lot in the clinical world, in the world of clinical trials. We have CROs that are specialized in this, is setting up labs, harmonizing the instruments, harmonizing the analysis and the data analysis to make sure we have repeatable samples from each of the clinical sites. But can we do one better? That's the question. Can we get back to where we do something for the samples to where we can get to this point? And that's kind of one of the things that my lab has been looking at and trying. The question comes up, all right, you're talking about live clinical samples. Well, why not just use PBMCs? And don't get me wrong, I love PBMCs. I think they're a great tool. We've generated a lot of fantastic data off PBMCs over the years. But they still suffer from a lot of the same things that our whole blood samples suffer from. Now, we've tried to have the clinical sites do the PBMC processing, and that's a bit of a nightmare. We have found that it's typically not uniform processing. Maybe not all the clinical sites have cytometry. Geez, see where my mind's been all day, right? Centrifuges or storage. And the other thing about PBMCs is when you're processing them into PBMCs, you are taking the cells out of their native environment. So they might not be the best sample for something like a receptor occupancy assay, or maybe anything where you want to keep the cells unmolested as possible. Also, you may lose some of your relevant cell populations. Here, we're only mononuclear cells are the only things that comes with PBMCs. And you'll still need to do the same kind of processing for the PBMCs that you would for whole blood assays, simply because they do have the same shelf life. Really, for PBMCs, from time you draw them to the time you can process them, really got about 48 hours. So you still have your stability issues. So what can we do? Well, there's a couple tubes on the market, and where's the laser pointer at here? Is that it? And I'm going to guess. That is not the laser pointer. Yeah, there we go. Maybe, yeah. So these guys, we probably use tens of thousands, maybe even hundreds of thousands of these guys in our life. This is the Cytometry BCT tube, blood collection tube. And basically what this does, it has a mild preservative in the tube. And it does tend to extend the stability of your markers. Now, if you're doing something as simple as, say, the IVD TB and K assay, you may have up to 14 days of stability from the time to draw to actually run your sample. Now, if you want to go deeper than that, typically you may get an extra day, maybe, of stability, but that's about it. And also, because the cells are fixed, you will see some changes from just say cells drawn into blood drawn into sodium heparin or EDTA tube. So there is another option, actually a couple options on the market. That's actually cryopreservation of whole blood. Will this help stability? Maybe. And this is from our lawyers here. I don't understand this one. They allow me to put the bottles up here, show you which bottles we use, but they made me block out the actual item numbers of the bottles or what the actual reagents were. I don't get it. That's BMS lawyers, fine, whatever. But if you want to just Google these two companies, and you'll be able to pick it up pretty quickly. All right. So we had more of this stem cell reagent in the lab. So this is what we started with. And what we did there is we took one of our lower order, very mature assays to do the initial testing with this. So this is a assay. It's a 11 color assay, but it's been ran on tens of thousands of immuno-oncology samples, and we know how it performs very well. And so we use this as our model assay. So what we've done and what we're looking at here is fresh blood from one donor versus cryopreserved. And the time point here, this is fresh versus cryopreserved for 24 days. So that's pretty good. And this is cryopreserved not in liquid nitrogen. This is cryopreserved at -80. We're testing -20 also, because even some clinical sites won't have a -80, but a lot of them do. Now, you see they're fairly, arguably, if you look at the scatters, there are different profiles here. These have been processed, preserved, and frozen. So the scatter profiles do look somewhat different. Major populations, they look almost identical. CD8, CD4s, looking at activation markers such as HLA-DR, CD38, even in the small populations of Ki67 positive CD4s, we see relatively similar staining. PD1 has always been a nice little smear in our hands using the antibody that we have to that's non-competitive for our drug. So when we look at this, just as a comparison here, this is from multiple donors. I'm showing 3 independent donors here, and these are normal healthy donors. It's not diseased, of course. These are just normal healthies. And what we've done is use this panel and look at either fresh versus cryo for 7 days versus 24 days. And you can see this is just showing the frequency of each subset here. And looks quite harmonious. And actually, when we look at the percent change from fresh, the dotted lines here, we're giving ourselves about a 20% change. Everything falls within that 20% change until you get down to the CD4 positive Ki67 positive cells. And of course, this is a rare cell population. We're dealing with very few cells in this gate, so that's more than likely what's driving this difference here. The same thing holds true at the 24 days of cryopreservation. Again, most of all our big populations are there with these smaller populations that are showing a little bit of difference. Now, this is great. We don't use this panel much anymore. So our next steps is actually put this into something a bit bigger, something on our Auroras, a 34 color panel, and that work is actually going on as we speak. We have used this also in one of our target engagement assays. Can we preserve phosphorylated proteins using this? And this is not as important for an assay where you bring the sample to your lab and then stimulate it. We have a lot of potential therapeutic agents that in the body actually drive phosphorylation signaling, right? And the PD biomarker here for these would be, can we take that blood out of a patient fresh, preserve it in a way where we preserve that phospho signal long enough to get it to a lab to test. And we tried this with, work. I think even the slides are tired. Give it a second. Oh, there we go. Okay, so we've done this with a small panel. Actually, these cells are stimulated with an IL-10 or a drug that's actually IL-10-like. Preserved, and this has actually only been preserved for 24 hours, but we look at T cells, CD4, CD8s, and we look at monocytes in this panel, both classical and CD15 positive monocytes. And then what we find is that after stimulation and immediate cryopreservation with IL-10, and to a lesser extent the drug that the potential drug, is we do see stimulation of course, the driving of the phosphorylation signal, and actually it is preserved quite well in the cryopreserved blood comparative across all times. Our drug here is actually more specific for monocytes than it is for T cells or B cells, and I won't go into why that is. I don't think I'm allowed to go into why that is. But also this one, looks like it will work for immunophenotyping and actually preserving precious phospho signal for clinical trials where that is important in the patient. Oh. Okay, now we can extend the ability of our samples. What do you think we ought to go after next? Anybody? Nobody read the full title in the book? All right, that's fine. That's fine, I'll tell you. So what we decided to go after next, maybe, go, is actually, come on, there we go. The human error part of it, right? So we're working on the big devil, and this is the next part of it. So right now in our lab, the putting together assays is still a very manual process. So what can we do to beat this human error, and why is this important? Well, this is our past, right? I told you about we have these 11, 12 color assays, and about 5 or 6 years ago, we thought these were huge. These were high order, and we ran tons of these things. Now today, we're all based on spectral analysis, spectral flow cytometers, running auroras, and we've combined all these down to 34 color assays. Well, almost everybody here understands that when you put together a 34 color assay, that's a lot of pipetting. And if you're running the proper amount of FMOs, that's even more pipetting. And there's a high likelihood that you could make an error in your making of your master mixes. So what can we do? Well, our first idea was we were developing an error detecting taser that would deliver a slight shock to the scientists of about 10,000 volts. Yeah, for some odd reason, the lawyers and didn't like that one again. Neither did our scientists. So I don't understand why. I see people out here taking notes, and I'm honestly a little worried about your labs. But after the see me, I've got blueprints. All right. Told you the jokes got way worse. Well, what did we actually do? We decided to automate some of the antibody cocktailing. Why is this a good idea? Well, it'll help maximize our workflow. It takes some of our hands away from actually getting carpal tunnel and pipetting out all these different master mixes and FMOs and whatever controls we need, and frees everybody up in the lab to do other things. And hopefully, if it works well, it should increase the accuracy of our assays and reduce any kind of human errors. So if we can do this, this is going to be great. We're going to preserve more of our precious clinical samples on our trials. If we can if it works well, we convince our other partner labs to do it, maybe we can get even further down the road to harmonization, not only thinking about instruments, but also assays. And of course, again, we're eliminating a lot of errors. It's challenging. It's not easy to do, especially when we're looking at the size of the volumes we're actually using our antibodies in. We need to be able to pipette very low volumes of antibodies to make the proper dilutions and do it accurately, do it over and over again, and do it with avoiding anything that could cause a problem with the assay, air bubbles, avoid cross-contamination, and it needs to be user-friendly. So with that being said, so we put together our idea, and we brought this over to our management. We said, okay, we need something we can easily put an input file in. We need a robot that can hold 4 degrees. We need to be able to have the number of antibodies be variable. We need to be able to calculate volumes pretty easily. It needs to be able to keep track of which samples are where, keep track of the volumes, and it needs to be able to understand, how not to do the cross-contamination, and we need to do stuff with less than 2 microliters. I said, great. I said, I think we can do this, but we need money. They said, you don't have money. But you do have an old TCAN. So we brought the old TCAN in, and thank God we had somebody on the at BMS, the guy name up there, Stephen, that was actually very capable in programming in the C language, because otherwise we would have been dead in the water, and helped us program this old TCAN up and get it running and kind of move to a proof of concept. So again, we did this We did this comparing our manual versus automated, and we did it using basically kind of the same assay that I talked about earlier. There was several different parts of phase one. First, we wanted to make sure it could do a serial dilution. We tried it on preserved products, varicels, basically lyophilized PBMCs. Then we tried it on normal healthy donors. And the issue of time, I'm just going to get to the punchline here of this. I showed you this assay earlier. It's still the same 11 color assay that we did for the test of the cryopreserved agents. And what we found is looking at each of the different donors that we have here. Blue dots is automation versus manual, and this is calculated in percent CV change. You can see quite harmonious for each of the reportables down here. Now, again, when you start seeing the spread a little bit, those are our rarer cell populations. But it looks like overall kind of proof of concept that we can run automated sample cocktailing or automated antibody cocktailing, and it works as good as our manual processes. So we passed all this. This is really where the rubber meets the road. Can this be used in the high order flow cytometry that we really wanted it for? And to do that, we tested it in a 34 color flow cytometry panel. We developed the programming error. So even developed the programming, we put it through its paces, did the same manual versus automated analysis. I won't belabor this. This is just what our panels looked like. There's actually two 34 color panels. We use the same 25 member core backbone, and also in one for an NK cell like panel and one for a T cell exhaustion or cytotoxic panel. And this is what the data looked like. It actually manual versus automated cocktailing. Of course, scatter works perfectly fine. These are not cryopreserved. These are all fresh. We have, everything looks good live dead. The lymphocytes look perfectly fine. We look for T cells. They match equally. B cells match really nicely. NK cells, again, match. Dendritic cells, we're getting down to these smaller populations. A little bit of change, but still within what we would consider stable or consider within variable limits for the assay. Monocytes and everything worked as good. So basically what this tells us is that we have a possibility of actually using this automated cocktailing in our 34 color panels. When we looked at several donors and applied the same 34 color panel to it, you can see these are the major, I didn't put every reportable up there. There's a couple hundred reportables actually out of these panels. These are the major ones with some very small cell populations, regulatory T cells and what have you. And what we found comparison is with being able to pass each of the reportables that are able to pass criteria for manual versus automation, which our criteria is less than 25% difference from manual to automation. Of course, all the very robust populations pass easily. And we had no less than 91.6% of all of our reportables passing. Again, giving credence to, hey, we made this work. We show this data to our bosses and they say, hey, that's wonderful. Now we'll give you the money for a new piece of equipment. We're testing something out new in the lab as we currently speak, or it's actually on its way. Hopes to be here next week. So just a little quick conclusion. Cryopreservation does seem to work. So looks like we can, even with at least a decently sized panel, we have up to 24 days stability cryopreserving it. Does work for phosphorylation. Where are we going next with this? Well, of course, we're going to apply this to our higher order panels, our 34 color plus panels. We want to see if it works for something along the lines of receptor occupancy. Probably not so much target engagement assays that would require activation of the cells. So we're taking this forward now, and we have shown that we actually can implement automation workflow, and it is suitable for higher order flow assays. It did alleviate, we believe it's going to alleviate a lot of stress in the lab. I'm going to try to keep as much stress in the lab as I possibly can because I don't like people getting, getting too comfortable. Just kidding, guys. I'm serious. The this did give us will give us a lot of increase of valuable time for our operators. And when we did a comparison, this is actually about twice as fast as manual preparation with high reproducibility. And hopefully we will have a reduced human error. And with that, thanks a lot for everybody. I appreciate you guys sticking around and listening.
Thank you. Thank you,
I did tell you I'd
Nathan.
keep it under an hour and a half. So there you go.
Thank you, Nathan. Any questions? We have plenty of time for questions.
I'm just curious on your take on the idea that, like, let's say that, that cytodelics, right? If I'm not mistaken, the buffer needs to be mixed with the blood, incubated, and then frozen. So what's your take on a clinical side being able to do that?
Yeah, that is going to be one of the challenges to overcome. So we'd actually have to partner with our CROs for that one very closely. CROs, for those not in the business, are the people that put together all the kits necessary for collect clinical specimens for our clinical trials. It could be done actually quite simply. We'd actually have to employ them to make tubes preloaded with the reagent, the cytodelics or the stem cell. And then it would just be a matter of pipetting in a couple mils of the blood after it's been drawn from the tube, something that can easily be done actually bedside. But yeah, actually bedside, as long as it's mixed well and gets to the freezer pretty quickly, it should actually be pretty feasible. Actually, quite a bit more feasible than producing PBMCs at a clinical site.
Any other questions?
I got you. Here, sorry, microphone for the Zoom. Thank you.
I think it's a wonderful thing to be able to automate the antibiotic cocktail production. I'm wondering if it would be an extra step that you would consider to maybe lyophilize and then be able to ship those cocktails pre-made to other sites.
Hey, lyophilization is something we've actually spent some time looking at. That was one of our methods we were thinking about doing. And it actually makes sense to a point. But when you're starting to get into a lot of, say, the covalent dyes like the BVs, and what have you, you run into problems with cross-linking there when you start drying them down. We've actually made it work with 10 to 12 color assays. We haven't really pushed it much further than that. So we talked to a couple vendors that can do it for us, and we actually had at one point or other time a dryer in the lab to try to do that, but we haven't really pushed it that hard, so I'm not sure.
And we actually have a question from Zoom. So James Dan is wondering, so what is your current lab policy for denying a sample that is too old?
What's that?
So what is your current policy for denying a sample that is too old?
Oh, so if we have something that's out of stability. So when we're putting together our assays, stability is one of the first parameters that we actually establish for each of the assays. So we know that each of the reportables will have its own independent stability. Some may have longer stability than others. Say if we have a sample where we have the majority of the samples have a 48-hour stability window. We have some that are 72. In our lab, what we'll do is we get a sample in that's beyond 72-hour stability. We will still run it so long as it's not clotted, right? If we can process a sample, we will process a sample and we'll report the data with a comment there saying, hey, out of stability. Now, arguably for a lot of the assays that we're talking about, this is in the exploratory realm, right? So these are not assays that are going to make treatment decisions on patients. These are to further the knowledge of the biomarkers for each of our compounds. So in that exploratory realm, then yes, we would run it and report the data with a comment saying, hey, take this with a grain of salt. Might not want to include it in the full analysis, but here's what we've got. Now, if we're at a level of a secondary endpoint or better, which is rare, but we do run some secondary endpoints, then that stability concern becomes even bigger. And we say if sample that comes in, majority of the stuff is out of stability, we will not run it. So then it becomes hard and fast cut.
Hi, Nathan. I got a couple. So how long how long would you say is an acceptable amount of time that it takes to make one of those 35 color cocktails? And have you looked into any stability of those cocktails? Like, so how long can you use it?
Oh, how long can you, like, after you stored it, after you made it and put it up in the refrigerator, right? Honestly, we don't do that a lot. We tried it in the past and we found, not the greatest results from that. After a while, like, we'd have to look at each independent cocktail, right? That's really, really what you got to do. Unless you're talking about, say, using a backbone that we could build, then we could really do those kind of studies. We haven't done that a lot with a lot of our panels because when we've seen things that fall off or we lose signal or what have you, then we just tend not to, jump the devil there. We kind of, let's just make it fresh each time, then we know it's okay. And God help us if we make a large cocktail and screw it up, burn all your antibody at one time, and making a lot of it, you made a dilution error, then you're being more of a mess than actually when you're making it fresh on a daily basis.
Yeah, we've had that same experience. All right, one last question. Have you looked into the sort of the next steps, the processing steps, and automating anything after staining your sample?
Yeah, that's probably next on our list. I think so. We tried this initially, and I think we bit off way more than we could chew, trying to go with the end-to-end process. Right? We spent time and a lot of money, and it honestly failed miserably. So we're taking the assays down and doing it in small bites. This cocktailing, that was a bigger bite. Next, maybe washing and spinning the plates or tubes, if that, but mostly we use 96-well plates. That may be the next one. Sample aliquoting to the wells would be another bite to take. I think if we go slowly and take this bite by bite, eventually we'll get to where we want to be and have maybe a mostly automated lab.